Tissue culture. Tissue culture is a technique of maintaining and growing plant of cells, tissues, or organs, especially on artificial medium in suitable containers under controlled environmental conditions. So, the term tissue culture is a process of growing cells, tissues, or organs in vitro or in a laboratory setting in which nutrient-rich media and sterile techniques. So, it involves taking a small sample of desired tissues, such as a leaf or stem, and placing it in a nutrient-rich medium under controlled conditions such as temperature, light, and humidity. So, however, the term tissue will then begin to grow and form a mass of cells which can be used for various applications, including plant breeding, genetic engineering, and drug discovery. So, why is it tissue culture is very important to the plant? We all know that the production of new plant from a small piece of tissue culture or cells removed from the growing tip of a plant in a suitable growth medium is called tissue culture or culture solution. So in this process, growth medium or culture solution is very important as it is used for growing plant tissue because it contains various plant nutrients in the form of jelly, or known as agar, and plant hormones which are necessary for the growth of plant. So one of the examples of tissue cultures are seed culture, embryo cultures, protoplast culture, and Callius culture. So seed culture is a technique used in plant tissue culture to grow plants in a laboratory environment using seeds as an explant. So a seed culture is a type of tissue culture in which germinating cells aseptically on artificial media, and the main purpose of seed culture is to increase the efficiency of germination of seeds that are difficult to germinate. So one example of seed culture is orchid. So the second one is embryo culture. Embryo culture is a technique for cultivating an embryo under aseptic conditions on a nutrient medium. So during this phase, fertilization occurs and the embryo grows and develops in a specific culture media within an incubator culture system in a combination of specific technical equipment and nutrients in the culture media that all IVF embryos need for optimal growth. So, the goal of embryo culture is to mimic the conditions of the female reproductive tract as closely as possible. So the third one is Callius culture. Callius culture is naturally developed in a plant as a result for a want. So the term Callius culture involves placing plant tissue samples in a culture medium supplemented with plant growth regulators such as auxin, cytokinin, and gibberellin to initiate callus formation or somatic embryogenesis. And the last one is protoplast culture. Protoplast culture are plant cells that have had their cell walls removed and they can be cultured to form cell colonies, ultimately leading to plant regeneration. So protoplast culture refers to the process of developing whole plants from the culture of cells without cell walls. So this technique, discovered over a century ago, is widely used in plant breeding and crop improvement programs. Protoplasts are cells without cell walls, and they can be derived from various plant tissues such as leaves, callus, cell suspension, and pollen grains. So, the general steps of a protoplast culture protocol involve plant material sterilization, isolation of protoplasts, and their introduction. Mericloning is the vegetative culture of plant for the tissue culture of apical meristems that began in 1960s. So, mericloning is a technique that used in plant propagation, especially in orchids, where a new plant is produced from the meristematic tissues such as shoot tips and buds of a donor plant. So these undifferentiated cells are grown in a semi-solid medium under sterile conditions to create new plants that are relatively identical to the parent plant. So the term mericlone is derived from the meristem cloning and refers to a plant that has been cloned via this method. It is a form of vegetative propagation that results in genetically identical plants. Mericloning has gained popularity due to its ability to produce large numbers of uniform plants. So, one of the examples of mericloning is orchids. So, these are the samples of mericloning. So, what is plant biotechnology? So, plant biotechnology is a rapidly growing field that has seen significant development in recent years. So, this technology is used to genetically modify plants to improve their traits, such as increasing their resistance to pests and diseases, improving their nutritional value, and increasing their yield. So, basically, it's all about genetically modifying plants to make them better. This can making them resistant to pests and diseases, giving them more nutrients, or even increasing their yield. So, let me break down some of the coolest recent development for the past 10 years. So, the first one is gene editing advances. So, gene editing advances is one of the most significant advances in plant biotechnology in the last 10 years has been the development of CRISPR-Cas technology. The second one is CRISPR-Cas. CRISPR-Cas is a powerful gene editing that allows scientists to precisely target and modify specific genes in plants. Being blessed with advances in CRISPR-Cas technology has been a game changer as a fancy gene editing tool that lets scientists precisely target and modify genes and plants. And the third one is engineering crop resistance. So engineering crop resistance, researchers have been able to create crops that can withstand pests and diseases without the need for pesticides. So a good example is a fungus-resistant wheat variety, which could be a huge deal for farmers. So another development in plant biotechnology has been the development of plants that are more tolerant to environmental stress and crop resistance. So crop resistance, this means that crop resistance refers to the ability to a plant to withstand or recover from damage caused by pests, diseases, or environmental stresses. Crop resistance is normally defined as the heritable ability of plants to escape attacking enemies partially or fully. And the fourth one is enhancing nutritional value. So enhancing nutritional value is another area of progress as improving the nutritional value of crops. Scientists have been able to develop crops that are essential nutrients such as vitamin A and iron. So one example of this is a rice variety that is rich in beta-carotene, which can be converted to vitamin. This could be a huge help in fighting vitamin A deficiency in developing countries. So the last one is enhanced crop yields. Enhanced crop yields is the practice that allows for taken together and biotechnology has seen significant development in the past years, with researchers developing new tools and techniques that have the potentials to improve their resistance to environmental stress, nutritional value, and yield based development. So that's all for today, and thank you.